Alright, welcome back. In this video, we're going to go over the Malate Aspartate Shuttle. The Malate Aspartate Shuttle is a super important topic in biochemistry. Professors love to ask the exam questions on it, and it's a high-yield MCAT topic. Now, do you remember in glycolysis, we made NADH+H. As a little refresher, NAD is an electron carrier, and the fully reduced form is essentially holding electrons. Now, in glycolysis, we made this. And I made a glycolysis video, so please check that out. But I mentioned that NADH+H is a currency, right? So this is a currency that we need to deposit into the bank. And that bank is the electron transport chain. So what will happen is when we deposit this money, in exchange, we're going to withdraw energy in the form of ATP at the electron transport chain. Now, here's the thing. Is we need to get to the bank. We need to be able to drive to the bank to make the deposit. We have the money and we know where the bank is, but we need a way to transport this currency to the bank. And that's where the Malate Aspartate Shuttle comes into play. Glycolysis happens in the cytosol, which is basically the outside of the mitochondria. The mitochondria is made up of the matrix and the inner membrane. The matrix is the very inner portion. That's where the Krebs cycle takes place. The inner membrane of the mitochondria is where the electron transport chain takes place. And that is where we need to make the deposit. Now, we need to get NADH+H into the inner membrane to make the deposit. But the problem is that NADH+H is not able to go through the mitochondrial membrane. It's not possible. Not with diffusion, nor with any channels or proteins. It's not possible. So that's why we use the Malate Aspartate Shuttle. Now, the Malate Aspartate Shuttle is basically a bridge or shuttle transportation system from the cytosol to the matrix. Now notice I did not say the inner mitochondrial membrane. That's where the electron transport chain is taking place. The matrix is where the Krebs cycle is taking place. So why are we doing this then? The reason is because this channel, this system, only works between the cytosol and the matrix, not the cytosol and the inner mitochondrial membrane. To get to the inner mitochondrial membrane from the matrix, that involves complex 1, which we'll get into in a later video when we go over the electron transport chain. But don't worry about it. This is like an intermediate step. So let's begin. So on the bottom here, this bottom section is the cytosol. This is where glycolysis is happening. The matrix is where the Krebs cycle is happening, but don't pay attention. Don't worry about the Krebs cycle. Just know that this area right here is like an intermediate step before we get into the inner mitochondrial membrane to deposit this NADH+H. So we're going to begin with aspartate here. So this aspartate is going to turn into oxaloacetate. Now the way this happens is a little complicated, so we're not going to really go over over that part because it's very advanced. But just in case if you were to be asked, or on the MCAT, how it works is alpha ketoglutarate, so alpha ketoglutarate, would turn into glutamate by an enzyme called transaminase. But there's also another name for the enzyme. But just really don't worry about it. It's not really that important. What's important is this part. The oxaloacetate will turn into malate using the enzyme systolic malate dehydrogenase. You must remember this enzyme. And we're going to use the NADH+H. This is what we got from glycolysis. We're going to take off the two H's, the two protons, and attach it to carbon number 2 here. So the double bond from oxaloacetate breaks. We are left with NAD+. And what's going to happen is this malate is going to go through this transport protein into the matrix freely. Now what's going to happen is the exact opposite. We're going to use the enzyme matrix malate dehydrogenase and essentially break off these two protons that we have here and reform or fully reduce NAD+ into NADH+H. And this can go to complex 1 now. Complex 1, you probably don't know what it is. That's completely fine. we're going to go over that when we get to the electron transport chain. But this right here is what will go into the inner mitochondrial membrane through complex 1. But now we regenerate, we basically get oxaloacetate now, and we go back to aspartate. So from here would be glutamate, and I'll get alpha ketoglutarate. Okay, and I believe it's the same enzyme, transaminase. And then this aspartate would go back down to this down the transport protein into the cytosol, and we would start this all over again. That's really it. It's pretty simple, right? It's kind of interesting though how a simple mechanism like this is. Well, it's actually kind of interesting that NADH+H is not able to cross the membrane. It's kind of strange. So we have to go through this entire process here just to get NADH+H into the other side of the mitochondria. So that's basically it. If you found this video helpful, please like and subscribe. Until next time, later.